Welcome to the part 2. In the previous part, we reviewed the structural details of interactions between SARS-CoV-2 receptor binding domain of the spike protein and the human cell ACE2 receptor. In this part, we are going to compare these interactions across different animal hosts and the corresponding infecting coronaviruses. The goal is to suggest plausible pathways which can be potentially utilized by the coronavirus to jump between species. In the application exercise, we will review the genetic diversity of ACE2 in humans, which might modulate susceptibility to the infection in different people. We start with the retrieval of the coding DNA sequence, or CDS, of human ACE2 gene using the NCBI gene database, directly accessible in MEGA. The retrieved CDS will be added to the MEGA Alignment Explorer. In the NCBI gene database, the name of the gene and the species are to be entered. As the next step, we have to inspect the returned list of genes matching your search query. We select ACE2 from Homo sapiens or humans. This brings up the NCBI entry for the human ACE gene. Again, we double-check the entry is indeed coming from humans. In the summary section, a brief description of the gene is provided. We have already reviewed the function of this gene in the previous part. The next section of the gene entry shows the genomic localization of the gene, highlighted in red. The next section of importance for us is the one showing the number of isoforms or transcripts the gene can produce. In the NCBI database, only isoforms or gene transcripts expressing protein products are shown. Our ACE2 gene has two transcripts, which are very close to each other, except that in the first isoform, one has untranslated exon at the start. In other genes, the number of isoforms might be larger, and some may undergo alternative splicing, resulting in essentially different protein products. The transcripts consist of introns, shown as the thin line, and exons, shown as the larger blocks. The exons, which are translated, are highlighted in darker green. Those which are not translated into amino acid sequence are shown in light green. Visual inspection of two ACE2 isoforms shows that the protein products produced by these transcripts are identical. Therefore, in the case of ACE2, we simply select the first transcript. The next section, important for us, collects the tissues and organs where this gene is expressed. The expression is estimated via abundance of the corresponding messenger RNA or transcripts per gene, sequenced by the method called RNA-seq. The number of the RNA-seq transcripts normalized by the length of the gene represents a proximal estimate of the amount of the corresponding messenger RNAs and the gene expression. For ACE2, we observe that the gene is selectively expressed in gallbladder, heart, kidney, duodenum, small intestine, and testes. Recent data published in 2019 show that it is also expressed in lungs. It is believed that the coronavirus primary attachment to these organs in tissue helps it spread through breathing, coughing via lungs, urination via kidney, sperm via testes, and excretion via guts. We have to scroll down to the next important sections containing links to the reference messenger and RNA sequences of all known expressible isoforms. Usually, each isoform has two links, one to mRNA starting with letters NM, and the other link to the expressed protein sequence starting with the letters NP. For our analysis, we are going to select the link to mRNA. Again, we double-check the name and the species of the mRNA entry. Note that the mRNA sequence corresponds to the processed isoform sequence, where the introns are spliced out. However, mRNA sequence still contains untranslated leading sequences, which we don't want to bring to Mega Alignment Explorer. To get only amino acid coding region of mRNA, we have to scroll down to the CDS link in the mRNA entry. Note the highlighted coding DNA part of mRNA and untranslated leading highlighted by a red rectangle. As a control, we shall check that the coding sequence starts with the ATG highlighted in blue or open reading frame triplet, common for all organisms. To download this coding DNA sequence, we select FASTA as the output format, and in a new window, we select FASTA as the text option. We double check that the sequence is starting with the ATG encoding open reading frame to prevent any surprises in the following sequence comparisons. From here, we locate and press the button Add to Alignment. The selected coding DNA sequence is added to the Mega Alignment Explorer. Mega Alignment Explorer, we change the long name of the sequence to indicate that this sequence of ACE2 corresponds to the human version. Next, we translate it into the protein sequence and check that the resulting amino acid sequence doesn't show any premature termination codons marked by asterisks and is of the expected length. In this way, we can retrieve and download all ACE2 coding sequences of species we want to compare. In this part, we would like to compare the virus binding interface between species of humans, pangolins, and bats. As the outgroup for the phylogenetic reconstruction, we are going to use frogs. We added dogs since in the application exercise we are going to address question whether dogs can be infected directly by bats and therefore can serve as the intermediate host for virus transmission from bats to humans. We use muscle alignment to align the selected coding DNA sequence using the Align Codons option. In the translated protein sequences, we immediately observe that frogs have the most divergent amino acid sequence featuring large number of insertions. However, in the middle section of the protein, the level of amino acid sequence conservation is high, indicating high homology of this protein across amphibians and mammals. In the next step, we are going to reconstruct and bootstrap the phylogenetic tree of this species using maximum likelihood method. This is the fastest method usually employed for a quick analysis of large genes or data sets. The maximum likelihood method suggests the position of the root, which in this case correctly placed on the branch leading to frogs. We accept this suggested routing since it does correspond with our routing using frogs as an outgroup. As expected, human and chimps are grouped together into the monophyletic clade representing primates. The pangolin sequence is placed closer to humans than bats or even dogs. That's yet another reason why pangolins were proposed as an intermediate host in coronavirus transition between all species. In the previous lecture, we established that only 8 amino acids of ACE2 form the interaction interface with the receptor binding domain of SARS-CoV-2. Therefore, we can extract these 8 amino acids from the key species of interest and compare how likely the interface from one species matches the interfaces from others. This may suggest possible pathways of cross-species transitions of the virus. For human ACE2, the 8 amino acids recognized by the coronavirus I put in the vertical column in the order of their appearance in the interface. The corresponding pangolin amino acids are extracted and aligned side by side with the human. The amino acids which are identical between human and pangolin interface are shown in black. The amino acids which are compatible with each other, like for example aspartic acid and glutamic acids, are highlighted in blue. However, the amino acids which are significantly different are highlighted in red. For example, change of histidine to serine reverses the electrostatic charge in the side chain and thus might result in the repulsion instead of attraction in the cognate amino acids. We can see that the bat interface contains more of the mismatched residues and is more divergent from the human one in comparison with the pangolin set. These observations result in the following transmission pathway explaining the natural origin of SARS-CoV-2. The virus endemic to bats with very different interface of ACE2 protein first jumped to pangolins, where it is evolved adaptations to the new ACE2 variant, which is closer to the human ones. Since so far viruses found in pangolins don't possess the furin cleavage site and they are in general more distant to humans than bat viruses, it was suggested that the virus jumped back to bats, where it acquired the furin cleavage site. From there it jumped either to dogs or from there to humans or directly to humans after somebody had eaten the bat in the Wuhan wet market. This pathway would require 4 or 5 critical jumps with significant underlying changes in the amino acid composition of the viral receptor binding domain and furin cleavage site. After each of the transitions, the virus must have undergone rapid adaptive evolution, acquiring a significant number of non-synonymous substitutions in comparison with normally occurring synonymous substitutions, as we studied before. However, rather different scenario of viral transition emerges if we look from the viral side. Here we list 7 amino acids from human infecting RBD. Note that asparagine 51 is repeated twice since it interacts with the 2 amino acids in the cellular ACE2. Here pangolin RBD interface is more different from human one, harboring 3 missense substitutions and 2 isomorphous substitutions. Note that on the bat side, out of about several thousands available genomes, currently only 2 bat genomes are publicly available, which can be assigned to the SARS-CoV-2 group. One shows 4 missense substitutions and a deletion, and is very unlikely to infect neither pangolins nor humans. The other one is famous bat rat G13 virus, discovered in 2013 and since then maintained in laboratory. Its RBD interface is actually very similar to SARS-CoV-2, with only 1 missense mutation of tyrosine 498. It's very likely that this virus may be able to directly infect human cells through multiple passaging through the human tissues. This tyrosine can be mutated to glutamine, making the virus fully compatible with human ACE2. Adding furin cleavage site in a lab would create SARS-CoV-2 out of progenitors of rat G13. Still, an outstanding question in this scenario is how similar are 3D structures of rat G13 and SARS-CoV-2. Of course, if structures are different, the different sets of amino acids can be exposed at the interface. However, modeling reported in this 2020 Current Biology paper shows that the 3D structures of RBD from SARS-CoV-2, pangolin and rat G13 are practically identical. This indicates that indeed the same set of the amino acids is employed by the virus to recognize its cellular receptor. And finally, one of the interesting arguments supporting the lab-made hypothesis of SARS-CoV-2 is near absence of evolutionary adaptation of the novel coronavirus to the new host humans. During the 9 months of the viral spread, the viral genomic sequences have diversified. This phylogeny shows an initial emergence in Wuhan, China in November-December, followed by sustaining human-to-human transmission, leading to sampled infections. There are about 100,000 of complete genomes available now, and the number increases by hundreds of every day. In this phylogeny, a small subset of only 3,000 genomes is shown. Let us look at the graph showing the number of acquired nucleotide mutations versus the time since the first genome sampled in Wuhan wet market. Most of the current variants have acquired no or very few nucleotide mutations. The largest number of the nucleotide substitutions is only 12 out of 29,000. The most of those are synonymous mutations, resulting in no change in the amino acids. A handful of those mutations, which indeed change the amino acids, are located in the less critical regions of the proteins, and none are found in the human receptor binding domain. This shows that the very first coronavirus found in the Wuhan wet market was already fully adapted to human host and had nothing to do with the animals sold in the market. The question remains how and where this adaptation had happened fully unnoticed if we reject the lab-made hypothesis. We conclude our lecture on the proximal origin of SARS-CoV-2 run by a short fragment of the investigation of the Sky News National Australia's cable satellite news channel.
Wuhan Institute of Virology, the laboratory now itself under the microscope. For months, the world wondered what really went on behind these walls. What's being hidden?
We do know that there was a lab working on these particular types of viruses in the immediate vicinity of the city of Wuhan. That's incontrovertible. If it did not come from the lab, that is one hell of a coincidence that it happened to come from a wet market not too far from the lab.
Researchers at the Wuhan Institute of Virology have been studying coronaviruses for years. Some of their experiments involve genetically altering viruses to make them more dangerous. Shizheng Li is one of their most senior scientists and a director at one of their labs. She's known as the Batwoman for her extensive experience working with the animals. She acknowledged the incredible risk of her work in a study she co-authored with the University of North Carolina in 2015. It stated,
"The potential to prepare for and mitigate future outbreaks must be weighed against the risk of creating more dangerous pathogens."
But her research got even riskier. Sky News can reveal, just 10 days before the first case of COVID-19, the Chinese government gave Shizheng Li's laboratory permission to engage in experimental activities with even more dangerous pathogens. This is confirmed in a statement from the director of the Wuhan Institute of Virology.
Wuhan National Biosafety Level 3 Laboratory was officially qualified for experimental activities and has the ability and conditions to carry out experimental activities of highly pathogenic microorganisms.
Xi's Level 3 lab is where most of the coronavirus research is actually carried out. So Xi was conducting higher risk research in a lab with lower security. The scientific view to date has been that COVID-19 did not come from this lab. But a growing number of scientists are coming forward, claiming that we can't rule this out.
We really don't know where this virus has come from. I mean, that's the truth. The two possibilities, which I think are both still open, are firstly that it was a chance transmission of a virus from an as yet unidentified animal to humans. The other possibility, of course, is that it was an accidental release of the virus from a laboratory.
US researchers suggest the virus may not have come from a Wuhan wet market. This study from Broad Institute of MIT and Harvard, awaiting peer review, states,
"Importantly, the market samples are genetically identical to human SARS-CoV-2 isolates and were therefore most likely from human sources."
And a paper written by Chinese researchers and published in the prestigious medical journal The Lancet claimed the earliest known COVID-19 case had no link to the wet market. They claim 13 people of the first group hospitalised also had no link to the wet market. Experts at Flinders University and La Trobe say two coronaviruses could have combined to create COVID-19 in a lab.
One of the possibilities is that an animal host was infected by two coronaviruses at the same time, and COVID-19 is the progeny of that interaction between the two viruses. The same process can happen in a Petri dish. In other words, COVID-19 could have been created from that recombination event in an animal host, or it could have occurred in a cell culture, experiment.
The full explanation of this is highly technical, but there are two key points. First, the virus binds more strongly to human cells than animals, including bats. Second, it is something its genetically closest virus in bats doesn't have, a technical term, what's known as a furin cleavage site, and that makes it significantly more infectious. Professor David Winkler says this all means that you can't rule out a lab as the source.
On the basis of the calculations we've done anyway, that you can't exclude that it's come from human, being processed through human cells in a biosecurity lab, but it's certainly not the only explanation.
There are many reasons to believe that the COVID-19 generating SARS-CoV-2 was generated in a lab, most probably by methods of genetic engineering. The method is very easy. Nucleic acid manipulation is a standard in many molecular biology labs.
Which pathway of viral transition, natural, zoonotic, featuring multiple jumps and requiring massive adaptive evolution, or a simple modification in a lab, are more plausible? It's up to you to decide. Thus, in these lectures, we've further extended and summarized facts and views contributing to the question of the evolutionary origin of SARS-CoV-2 virus. We practiced the skills of the genomic sequence retrieval, alignment, phylogenetic reconstructions, and analysis. In the application exercise, we will review biodiversity of human sapiens. This will help to answer the question whether in divergent human populations a mutated allele of ACE2 may exist, which might render the carriers immune to COVID-19. Thank you very much for your participation in this course. We have practiced retrieval, graphical rendering, analysis, and prediction of structures of biomolecules, primarily proteins. We reviewed and practiced some basic methods of comparison of nucleotides and amino acid sequences across related proteins. Of course, we just scratched the surface of this mighty and rapidly developing field of informational biology. These skills might empower and enhance your own research or lower the barrier in your use of the vast amount of biological information freely available online. Nevertheless, I hope this course might contribute to the success of your life story. I am Konstantin Perlushin.